Hello students, welcome to this video. In this video lecture, we are going to talk about key answers of Neet 2024, which was conducted on 5th May, Sunday 2024, that is today. So the paper that we are going to discuss is T3 booklet. So from T3 booklet, botany questions we are going to discuss. So we'll just move on, scroll down to the botany part. This year paper was very easy, damn easy, especially the biology part. Easily within 40 to 45 minutes, you can solve all the questions. All were direct. And all the questions were from NCERT. So there is no question outside NCERT. So we will discuss as we are going to talk about all the other questions. So we are starting from botany 101. Identify the set of correct statements. The flowers of Vallisneria are colorful and produce nectar. Vallisneria is an aquatic plant. Aquatic plants are water pollination, hydrophily. So for Vallisneria, they don't have to produce any fragrant color or beautiful flowers or fragrance or nectar. So this statement is wrong. Flowers of water lily are not pollinated by water. Yes, the flowers of water lily and lotus, they all are not pollinated by water as an agent. They are pollinated by wind, anemophily. So statement B is correct. So option B is correct. So statement B is correct. In most of water pollinated species, the pollen grains are protected from wetting. Very true. In water pollinated species, what happens? The pollen grain is always released into the water medium. When these pollen grains are always in contact with water, there's a high chance of getting desiccation and all those things, and they might be so get spoiled early. So what happens? They're always covered by waxy coating. This waxy coating helps from desiccation, dehydration, etc. So statement C is also correct. Pollen grains of some hydrophytes are long and ribbon-like. Yes, we have also seen in Vallisneria and other Zoostera and all. Pollen grains of hydrophytes are long and ribbon-like. Very true. In some hydrophytes, pollen grains are carried passively inside water. They're released within the water. They are not on the superficial medium. Within the water medium, they are carried forward and they reach the stigma. So B, C, D, E. All the statements are correct. Only A is wrong. So the option 2 is the right answer. You can eliminate option A. So 1 and 4 becomes wrong over there. So the correct statement is going to be option 2. Moving on to the next question. The type of conservation in which the threatened species are taken out from their natural habitat. So it's removed from their natural habitat. If they're protected in the same habitat, it's called in situ. When I remove it and place it in some other zoological parks or botanical gardens, I'm removing it and placing in some other way. It is called as ex situ conservation, to be more specific. Since X8 is not given, we can go with this option called biodiversity conservation, the most appropriate one. So the actual one should be for XC2, but since XC2 is not there, we are going for biodiversity conservation. Direct question. Inhibition of succinic dehydrogenase enzyme by malonate is a classical example of competitive inhibition. It's from Biomolecules chapter. So succinic dehydrogenase enzyme goes and binds to the succinic acid. This succinic acid and malonic acid, they are structural analog to each other. They both closely resemble to each other. So if succinic acid is not there, then malonic acid can easily go and bind to that enzyme and it inhibits the binding of succinic acid. Structural analog, we use a word. So it's an example for competitive inhibition. So succinic acid and malonic acid are going to inhibit by competitive inhibition. So the answer is going to be competitive inhibition. Next one. Identify the part. This is the where most of the people have gone wrong in this. The reason for this is you have not seen the question properly. Identify the part of the seed from the given figure which is destined to form root. The question is you're going to form root. So which forms the root over there? Just a minute. So which is going to form a root over there? See, option C and D is there. D is nothing but coleorhiza. Coleorhiza is just a protective covering. protective covering which covers the radicle. C is the radicle. So in morphology chapter also we have studied this. So radicle and plumule, it is the radicle that gives rise to the root system, plumule that gives rise to the shoot system. So our answer is 1. So it is radicle that is going to give rise to root system. Bulliform cells, such easy thing in anatomy we have studied. Some epidermal cells are becoming colorless, round, circular, called as bulliform cells or mota cells. And these are present on the upper epidermis. They are helping in rolling the leaf inwards. When the leaves are rolled inwards, and this happens when the water content is less, when temperature is more. To avoid transpiration, leaves are rolled inwards, which is seen in only monocots, exclusively monocot. So they are curled inwards in monocots to reduce the rate of transpiration. So it's option 3. Which of the following are required for dark reaction? In photosynthesis, we have studied light reaction and dark reaction. Dark reactions are independent of light. Light is not required. So A is not required. Chlorophyll is a pigment. See, it requires chloroplast. In chloroplast, stroma, the matrix is the region where dark reaction takes place. So chlorophyll is also not required. Chlorophyll is just a pigment which is required for light reaction, not for dark reactions. So carbon dioxide, definitely carbon dioxide is reduced to form sugars, ATP and NADPH. ATP and NADPH is required. So it's option 1. C, D, E only is required. Formation of interfascicular cambium from parenchyma cell. So parenchyma cell is a permanent tissue. From permanent tissue, interfascicular cambium is going to be formed. Interfascicular cambium is a meristematic tissue. Permanent tissue to meristematic tissue is dedifferentiation. Later, this interfascicular cambium again gives rise to secondary xylem and secondary phloem, which is the permanent tissue, that is redifferentiation. So this again it goes and forms redifferentiation, that is not required. So the answer is dedifferentiation. Very easy direct questions. Hind to eco R1, these are all restriction enzymes. Restriction enzymes cut at a particular sequence called as recognition sequence, which are palindromic in sequence, and usually they are 6 base pairs. So the answer is 6 base pairs. For Hind to eco R1 also GAATTC. All the sequences in biotechnology chapter you have studied principles and processes. So direct question again. Tropical regions show greatest level of species richness because tropical latitudes have remained relatively undisturbed for millions of years. Hence, more time was available for species diversification. True, tropical environments are more seasonal. B is wrong. Tropical environments are not seasonal at all. They are less seasonal. So, see the option where B is not there. The only option where B is not there is 3. So it's going to be option 3. More solar energy is available, constant environment, tropical environments are constant and predictable. Because of this, what has happened? More number of species, diverse species has been obtained. Species richness is seen. So it's option 3. Moving on to question number 110. Which one of the following is not a criteria for classification of fungi? From biological classification, this is the only question that has appeared, I believe. So, mode of spore formation, yes, whether it is a zoospore or ascospore or basidiospore or endospore or exospore, whether it is formed endogenous or exogenous, this has been taken into consideration. Fruiting body, yes, whether it is ascidium or basidium, ascocarp or basidiocarp, that is also taken into consideration. Morphology of mycelium, phycomycetes is cenocytic aseptate, while our basidiomycetes, ascomycetes, deuteromycetes, all these three are septate. So morphology is also taken into consideration. Nutrition is not taken into consideration. All are saprotrophic, all are heterotrophic. It is either parasitic or saprotrophic. So no mode of nutrition is not taken into classification of fungi. So it's option 4. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide? Very, very easy question. For the carbon dioxide to be fixed or to form glucose, you need 18 ATP molecules and you need 12 NADPH molecules and you need 6 carbon dioxide. For 1 carbon dioxide, it becomes 3. Just divide by 6. 6 3s are 18, 6 2s are 12. So you require 3 ATP molecules and 2 NADPH for 1 carbon dioxide. It is required. Calvin cycle. Again, it comes under your photosynthesis. Another very important topic. These are regarded as major causes of biodiversity loss. So it is very, very important. Mutation is not there. Habit loss and fragmentation is there. Co-extinction is there. Over-exploitation is also there. And A, B, and D. So in this case, what the major causes of biodiversity loss, over-exploitation, co-extinction, habitat loss, and you get your option 2, A, B, and D. So migration is not going to cause biodiversity loss. It's just change in biodiversity. The biodiversity does not lose over there. So it is going to be just A, B, and D. Option 2. The capacity of to generate a whole plant from any cell of the plant is called. So this actually is a part of your strategies chapter, strategies in production, food production. But now what has happened in new NCRT, they've taken a portion of your strategies chapter and they have incorporated in biotechnology application itself. So it comes under tissue culture, where from a single cell, the whole plant can be regenerated using tissue culture. And the principle is totipotency. For animals, we use a term called as stem culture, stem cells. Here it is called as totipotency, where they can differentiate and give rise to your entire plant body in tissue culture, totipotency. The equation of Verhulst per logistic growth. So this again comes in organism and population. K stands for carrying capacity. Spindle fibers are attached to kinetochores of chromosomes. If you look at your chromosome structure, to the centromere, the kinetochores are attached. Kinetochore is a protein that is present in the centromere. To this, the spindle fibers are going to be attached in metaphase plate. After they are attached in metaphase plate, they are pulled apart. So it is seen in metaphase. So option is going to be metaphase. Spindle fibers are attached to metaphase part. This is the direct NCRT question where A and B both are perigynous. So both are perigynous and perigynous. Option 2, where they are attached in the rim of the thalamus. All the floral parts are attached in the rim of the thalamus. The one common thing I have seen in all the match the following option, if you know one option correctly, one answer correctly, you can easily find out the answer. Now, rhizopus is bread mold. A is 3. So then Eustilago is mud fungus. Puccinia is going to be, Puccinia is rust fungus. And Agaricus is mushroom, Basidiomycetes. So it's option 3. It's going to be option 3. Next. In a plant, black seed color is dominant over white seed color. So black seed, it can be either homozygous or heterozygous, and it's a dominant feature. Its recessive is white color. In order to find out the genotype of the black seed plant, you have to do backcross. In backcross, we will always take recessive parent. So it's option 4. We are going to take the recessive parent. A pink color flowered snapdragon. Snapdragon is is an incomplete dominance. It is going to show. So incomplete dominance was crossed with a red flower. They have not said whether it is homozygous or heterozygous. So okay, it can be RR homozygous condition. What type of phenotype is expected in the progeny? When RR is crossed with RR, you can also get it's crossed with pink type, right? Yeah, pink type, you're going to cross it. If you get capital R, you will get red flower. If you get capital R, small r, if it is an heterozygous, you will get pink flowers. So white flowers will never come. Only pink will not come and only red will also not come. So it's going to be option 4. It's an incomplete dominance. That is the reason. Two or more alternative form of a gene is called as allele. If you're crossing F1 with recessive parent, it is called as test cross. If you're crossing with any of the parent, it is called as a number of, cross with the recessive parent is called as, test cross. With any of the parent, you're calling it as back cross. The number of chromosome set is called as ploidy. If it is having one set, haploid. 2 set diploid, 3 set triploid. So it is called as ploidy. So with this you get with option 1. You go with option 1. Lecithin, biomolecule. It comes under phospholipids. See, it's so easy. You don't even have to read any full statement. Clostridium butylicum, butylicum microbes in human welfare chapter. Butylicum gives rise to butyric acid. Saccharomyces, yeast is used for ethanol production in case of industries. Alcoholic beverages, we are using yeast. Trichoderma polysporum, cyclosporin is used. Streptococcus, streptokinase is used. It's option 1. Direct question. If you know any one, you can answer this easily. This is another very good question. In the given figure, which component has thin outer wall, a thin outer walls and highly thickened inner wall? So it is your guard cells which is having thick inner wall. The inner wall is thick and outer wall is thin. Guard cell is nothing but option C. C represents guard cell. So it's option 3. Which of the following is an example of actinomorphic? Pisum, Sesbania, Cassia. They all belong to Fabaceae. Fabaceae shows zygomorphic flower because they have papillonaceous corolla, 9 plus 1 arrangement. So they are zygomorphic in nature. The only odd one out is Datura, which belongs to Solanaceae. So answer is Datura. A transcription unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream. You have promoter region towards 5' and structural gene terminator towards 3' and downstream region. So it is option 2. What is the fate of a piece of DNA carrying a gene of interest which is transferred into an alien organism? Biotechnology. When a piece of DNA, foreign gene, alien gene, gene of interest, it is being inserted into the host organism, it cannot multiply on its own. So option A is rejected. It may be integrated into genome or it may not be. So option B, statement B is true. It may multiply or it may not multiply. Again, C is true. Alien piece of DNA is not an integral part of chromosome. True. The piece of DNA what we have added is not an integral part of chromosome. But there is no option where B, C, D is present. So the most appropriate one is going to be B and C. Then it's option 1. If B, C, D was there, then you could have gone for D. Because the alien piece is not an integral part of chromosome. After it is entering, it becomes an integral part. So there's some ambiguity over there, but there is no option where BCD is there. So you can close your eyes and go for option 1. Auxin 2,4-D is used by gardeners to prepare weed-free lawns, but it is only selective. It will kill only the dicotyledonous weed. It will not cause any harm to monocotyledonous plant 2,4-D. So it's option 1. The factor of the enzyme, the cofactor of the enzyme carboxypeptidase is zinc. Zinc is as a direct one, no, nothing to think. The lactose present in the growth medium of bacteria is transported to the cell by the action of permease. Polymerase does not come at all. Transacetylase helps in lactoperon, metabolism of lactose. Beta galactosidase breaks galactose into lactose over there. So the answer is going to be permease. It allows the permits. It allows the entry of lactose. Which one of the following can be explained on the basis of law of dominance? What did Mendel say? According to Mendel's law, one of the pair is dominant, the other one is recessive. True. Alleles do not show any expression, and both the characters appear as such in F2 generation. Wrong. Both the characters will not appear. It's only any one character, either the dominant character is going to appear, or if it's an heterozygous recessive character is going to appear. So B is wrong. Factors occur in pairs in normal diploid plants. Yes, why it is appearing in pair? One is from paternal chromosome, the other one is from maternal chromosome. So it is always in pairs. The discrete unit is called as factor according to gene. According to Mendel, later Johansson named it into gene. Expression of only one of the paternal characters found in monohybrid cross. True. So A, C, D, E. Is there any option? Yes, option 4. A, C, D is the right answer. This is very good question. Direct NCERT statement where people will get confused because the other genes are not given. Bt toxins are insect group specific and coded by a gene CRY1EC. They have not said only gene. So this gene also codes for cry protein. So this statement is correct. Bt toxin exists as an inactive protoxin Bacillus thuringiensis. However, after ingestion by the insect, the inactive protoxin gets converted into active form due to alkaline pH, not due to acidic pH of the gut region. What happens in the gut region because of alkaline region? Alkaline pH, it gets converted to toxin crystalline. It goes and binds to the midgut epithelial cell, oozes out, creates a pore, and because of endosmosis and ecosmosis, blasts and the organism dies. So statement 1 is correct, 2 is wrong. Which is the answer? Statement 1 is true, but 2 is statement 1 is true, but 2 is false. So it's answer 1. 132. Parenchyma is a living, but collenchyma is dead tissue. Wrong. Parenchyma and collenchyma both are living. Only dead tissue is sclerenchyma. So that is wrong. Statement 2. Gymnosperms lack xylem vessels, but xylem vessel is a characteristic feature of angiosperm. True. Gymnosperm has xylem tracheid. They lack xylem vessel. And angiosperm has xylem vessel. That is a characteristic feature. So statement 2 is correct. 1 is wrong, 2 is for 2 is so it's option 2. Statement 1 is false, 2 is true. Chromosomes become gradually visible under microscope during leptotene stage. After packeting leptotene, it becomes visible. True. Statement 2, the beginning of diplotene stage is recognized by the dissolution of synaptonymal complex. What happens in diplotene and diakinesis? Terminalization of chiasmata and the synaptonymal complex is going to dissolve. So this is also true. Both the statements are true. Answer 3. Centriole shows cartwheel 9 plus 1 organization. Centriole, I'm sorry, 9 plus 0 organization. It is going to show cartwheel like it has outside. It is going to have triplets. 9 microtubules which are arranged in triplets. Cartwheel organization is true. Nucleolus region is associated. It's found. It's associated and it's an active rRNA synthesis. Leucoplast is a type of plastid colorless where it is going to store nutrients. Golgi apparatus is a site where glycolipids and glycoproteins are going to be formed. So it's going to be option 3. Look at this question. Even an 8th standard kid can answer such easy question. List of endangered species was released by IUCN in Red Data Book, International Union for Conservation of Nature and Natural Resources. The DNA that is present in chloroplast, the DNA semi-autonomous organelle, let it be chloroplast or mitochondria, it is circular and double-stranded. Which of the following are fused in somatic hybridization? Your potato and tomato was fused to form potato and tomato. So by protoplasts. This flower is nothing but it's not cleistogamous. Cleistogamous will be closed and it is not a compact inflorescence. It is definitely wind inflorescence, wind impollinated. It's not water pollinated with an well exposed stamens. So it's option 3. Spraying sugarcane with gibberellins, the fold 20 times the increase was fold was crop yield was increased by 20 fold by spraying gibberellic acid. Bolting internode elongates, yield also increases. So direct question from plant growth and development chapter, easy question. Frederick Griffith was the one who was involved in transformation. Jacob and Monod for Lacoperon, they were awarded Nobel Prize. Hargobind Khorana played a major role in genetic code. Messelson and Stahl in semi-conservative mode of DNA. So it's option 4. So easy. Almost 25 to 30 match the following questions had come so easy this time. GLUT4 is nothing but it allows the transport of glucose. Insulin is an hormone. Trypsin is an enzyme. Collagen is an intercellular ground substance. So again, option is going to be 3. This question is nice. Again, in photosynthesis, in C3 plants, some oxygen binds to rubisco, hence carbon fixation is decreased. S. Rubisco, ribulose, bisphosphate, carboxylase, oxygenase. It can bind to carbon dioxide also. It can bind to oxygen also. It depends upon the carbon-oxygen ratio. If oxygen is more, it binds to oxygen and reduces the photosynthetic yield, which is known as photorespiration. Respiration. So the statement is correct. Statement 2. In C4 plants, mesophyll cells shows very little photorespiration. Bundle sheath cells do not show respiration. In C4 plants, mesophyll cell shows zero respiration, photorespiration, because mesophyll cell lacks rubisco. Chlorophyll dimorphism is seen over there. Rubisco enzyme is absent. So zero photorespiration. So statement 2 is wrong. So is there any question like that? Yes, option 1. Statement 1 is true, but 2 is false. Identify the step in tricarboxylic acid Krebs cycle. They're talking about TCA cycle or Krebs cycle, which does not involve oxidation. Always remember, always oxidation and reduction is coupled. Whenever oxidation, loss of electrons, loss of protons is taking place, that is taken by NADP plus and you form NADPH. So it is reduction. NADPH is going to be formed. This is reduction. Wherever oxidation takes place, reduction also takes place simultaneously. In which step NADPH is not formed? That is the answer. In all these three, NADPH is formed. This is the only step where NADPH is not formed. So no reduction, no oxidation. So it's option 1. Direct question. Citric acid cycle, Krebs cycle, TCA cycle occurs in mitochondrial matrix. Glycolysis occurs in cytoplasm. Many times I've said for my students, electron transport system, it takes place in the inner mitochondrial membrane. Proton gradient is towards the space. So it's going to be option 4. Which of the following statement is correct regarding the process of replication? In replication, always DNA polymerase undergoes polymerization in 5' to 3' direction. Why 5' to 3'? In 3', there's a free hydroxyl group, so where your new DNTPs can be added. So it's always option 2. Next. This is just 10% law. It looks like it's very complicated, but in what is happening in the net primary productivity is 100. So for the next second trophic level, you'll get only 10, and the next trophic level, you get only 1x. 1x is there only in 4, so it's going to be option 4. Mango is true. Cotton is twisted estivation. Pea is marginal placitation. Rose is an example for perigynous flower. 3. Today this year's paper is very easy. There will be definitely many people who are scoring 360 on 360 and 720 on 720 this year. Many people, because so easy biology and zoology has become. Alexander von Humboldt, he was the one who gave species area relationship. Robert May was the one who gave global species diversity at about 7 million. Paul Ehrlich was the one who gave Rivert-Dropper hypothesis. David Tillman, long-term ecosystem. So it's going to be option 4 again. Very easy. 149. Oh my God. So many direct questions. Monoadolphus, single stamen. All the stamens are fused to form a single bundle, which is seen in hibiscus, China rose. It's a characteristic of Malvaceae. There has been no questions outside NCRT and no question from other concept also. Diadolophus, 2 condition 9 plus 1, which is seen in P. Polyadolophus, many bundles seen in citrus. Epiphyllus, where stamens are attached to tepals, which is seen in monocot, lily. So it's going to be which option? Monocitrus, Polyadelphus C1. There is only one option where it is C1. It's going to be 3. Read the statements and show members of Phaeophyceae. In Phaeophyceae, sexual reproduction is by oogamous method only. Sexual reproduction is by iso, aniso, and oogamous. So B is wrong. So any option where B is there, it's going to be eliminated. B is there, B is there, B is there, eliminated. You land up to only one. You don't even have to read the other statements. See, so easy this year it is. Time conservation also is there. So zoology, these are all classical zoology, which I will not be discussing. Let me see if there's any common questions which is there, like molecular biology or genetics. I will discuss along that. Yes, 157 RNA polymerase 3' to 5'. So 3' to 5', the polymerase is going to become 5' to 3' characterization. So question number 157, it is going to be option 3, where if T is there, A will come. If A is there, T will not come. It's an RNA. RNA polymerase is involved in transcription. So from DNA, mRNA is going to be formed or RNA is going to be formed. So instead of T, you will get U. C is there, G will come. A is there, T will not come. Again, U will come. Like this, you can go on doing it. You will get option 3. Next, we will go for this cell cycle. Yeah, in cell cycle, the first one is E. Then you're going to get for after gap one phase, S phase, synthesis phase, where DNA replication. Then you're going for A, G2 phase. Then you get cytokinesis. Then you get karyokinesis. E, C, A, D, B. There's only one option, option 2. Lipase, nuclease, protease is an enzyme that breaks protein. So it's going to break peptide bond. Amylase is an enzyme that is going to break your glycosidic bond. If you know these two options itself, you can go for option 1. Then what else is there? Hmm. Yeah, another biotechnological question. Enzyme alpha 1 antitrypsin is used for emphysema to treat emphysema related to your bronchitis, cough related disorder. Cry 1AB, cry 1AC. Forgot it, don't worry. Enzyme replacement therapy was done on a 4-year-old girl who was suffering from SCID, severe combined immunodeficiency, or adenosine deaminase deficiency, using gene therapy, biotechnological application. If you know this two itself, you can mark option 1. Another very easy direct question. Yes, coming on to this. X is nothing but ORI site. ORI site helps in replication. Y is nothing but ROP. It is helping in it helps in protein that is required for replication. So gene X ORI, the stronger the ORI, more the copy number. So it's option 4. Y is ROP, proteins for replication it is involved in. Next question. Okay, we have another cell chapter. Axoneme. Axoneme is there in cilia and flagella. Axoneme. Cartwheel-like structure is found in centriole. What is axoneme, cilia, and flagella? It is coming under there. Then crista is found in mitochondria. Inner membrane of mitochondria has several infoldings known as crista. Satellite is a part of chromosome. Apart from nucleolus, secondary constriction, or the remaining portion is called as satellite chromosome, satellite body. So it's option 2. Which of the following is correct? Incorrect. Sorry. Which of the following is incorrect? Bioreactors are used to produce small scale, large scale. So this is incorrect. So this will be answered. You don't have to sit and read the remaining portion and waste your time. Diakinesis, terminalization, packaging, recombination, nodule recombination takes place over there. Zygote, two homologous chromosome pairs of synaptonymal complex is going to be formed. Leptotin chromosomes are looking like threads. So it's going to be option 1. Direct option 1. TI plasmid, tumor-inducing plasmid in Agrobacterium tumefaciens, which causes crown gall disease in case of your plants. So that is again TI stands for tumor-inducing. Direct, so easy thing. This time people should definitely get out. So many things are correct. Many things are very easy and direct over here. 185 again genetics. 185 is again option 1. Klinefelter is seen in X chromosome. Additional of X chromosome, it's seen in only males. Klinefelter syndrome. So again, easy. Then anything else is there? One more very easy thing. Stata box is there in promoter, and termination occurs because of rho factor. And with this, you have only one option where 190 is option 2. Stata box comes in promoter region. So these are all typical zoology, which will not be discussing. I think, yes, one more question. Gauss competitive exclusion principle states that two closely related species competing for same resources cannot coexist. If they're competing for different resources, they can exist. So statement 1 is wrong. I'm sorry, statement 198, the answer is going to be 198. What happens over here? Statement 1 is wrong. According to Gauss principle, the inferior will be eliminated, true, if the resources are limiting. So it's going to be option 2. Regarding the catalytical cycle of an enzyme action, what happens? First, what is going to happen? The substrate is going to bind to the active site of an enzyme. So E is the first thing. Second, this substrate is going to bind with enzyme and form substrate enzyme complex formation. Second one is E. There's only one option, so it's going to be option 3. ABO blood group system. Father is B positive, mother is A positive. And if the child is O, O is recessive. It comes only if the parents are heterozygous. So parents should be IBIO and mother should be IAIO. Then only IO and IO can come together. So it is going to be which option then? IBIAI. So it's going to be A. Option 3 only. And 200. Mitochondria and chloroplast both are double membranous structure. So statement 1 is correct. Both are double membranous organelle. They have outer membrane and inner membrane, and they also have perimitochondrial space and perichloroplast space, periplastic space. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Let it be mitochondria or chloroplast, inner membrane is less permeable. So it's going to be wrong. So it is going to be option 1. Statement 1 is correct, but statement 2 is incorrect. So it's going to be option 1. So with this, we completed the biology discussion. So according to this analysis, biology paper is very, very damn easy. And outside NCERT, there has not been any question, no ambiguity, no tricky questions, direct answers you can get. And within 40 to 45 minutes, you can solve. And 360 on 360 is possible in this paper. 720 on 720 will be the first rank this time. So it's very damn easy. I hope this discussion was beneficial for you people. I really thank you for listening to this my video lecture, and please, if you find this video beneficial, please share this video link to your friends and other classmates, and I wish you all the very best. And for those who have didn't score and those who are really determined, please take a career option. If you're really passionate into medicine, then you can definitely enter into medicine field with 1 year gap. So you can come and join repeaters batch, and repeaters batch take a 1 year program. In 1 year program you can definitely get into this. For those who are in the border range, for those who are there around 400 450, definitely if you are a general merit candidate you will not get medicine seat. And if you are there in above 400, definitely if you take 1 year break and if you join any repeaters course, any full-time course, definitely you can get into medicine seat, medical seat next year, and you can become doctors. So my sincere advice would be like, for those who are there, just a minute, let me just minimize this and let me just show you my screen. Okay, forget about this. So now there are many institutes which has certain full-time repeaters course. So for those people, for those students who are there in and around Karnataka or Bangalore, so I was working in previous ex college called base educational services base college, which is there in Basangudi. So they offer full-time course. Or if you are there in any part of Tamil Nadu, here currently now I'm working in Allen Institute, Allen Career Institutes in Tamil Nadu in Madurai branch. So you can approach me. My email ID I will be giving. It's rakeshb96@yahoo.com. RAKESHB, B for Bangalore, B for ball. rakeshb96@yahoo.com. You can contact me for any of the admission queries. So I have been associated with the Base College in Bangalore, Allen College at Madurai. So if any kind of help or support you want for admission, I can help you forward. So if your score is above 400 or 350, 400, definitely with 1 year coaching, 1 year break, you can enter into medical stream. So don't lose your hope and confidence. If you want, you can get in touch with me. We can discuss what can be done, and I can guide you for other career related issues, career related guidance also, which will be done for free of cost. So I hope you were all benefited with this video. Please do share this video with your friends and other members. Thank you. Wish you all the best. We will see in other next video lecture. Do like my channel, subscribe to my channel, and subscribe and share this video to everyone. Thank you.